Okay, here we go. Atoms to molecules to organelles. In school, we spend a lot of time talking about atoms, atoms, and molecules and organelles, but we sometimes forget that they're all part of a bigger picture. And we don't often teach everything all sort of together. So you don't get a feel for how one thing becomes another. You learn about atoms in grade 9 and you draw Bohr-Rutherford molecules, or Bohr-Rutherford diagrams in 2 and 8 and call this, something, and protons and neutrons, and you draw these pictures. And then all of a sudden, the next thing you know, you're drawing a cell, and you're drawing a thing like this, calling it a mitochondrion, and a nucleus, an endoplasmic reticulum, and a membrane. Well, there's no connection. How do these atoms actually form these structures? How does it all sort of fit together? That's what we want to look at now, and we want to build towards an understanding of this special DNA molecule that is so important in genetics. So, we begin with the fact that atoms have a job. And their job is just to fill their outer orbits. Remember grade 9. And the reasons for this are very complicated. The reasons for this are found deep inside the nuclear physics of atoms and how atoms sort of obey the fundamental laws of electromagnetism. And that's a very complicated thing, and it takes many years to understand it. So we simplify it by saying atoms want to fill their outer orbits. And what we say is that somehow when they do fill their orbits, they become more stable. In other words, this is sort of a state that all of those forces that I talked about are sort of in balance. And that's what we want, balance. So without getting into the deep physics of it all, we understand that atoms want to fill their outer orbits so that they can become stable, which means that all of the fundamental forces that are pushing and pulling and twisting and moving and doing their thing are all in a sort of a state of balance. Okay? So you can think of stable as also being balanced. Now, how do atoms do this? They do it in a number of ways. But essentially, what they do is they can exchange electrons by giving them to each other that are in their orbits. We call this ionic bonding. So you learned all about this in grade 9 and 10. How atoms give electrons one to the other, they become ions, positive and negatives attract, and you get things like, sodium chloride and calcium sulfate. That's not going to be, let's make it calcium nitrate. So I can use my bracket that I draw, calcium nitrate. And we learn how to do all these formulas, and we do all the crisscrossing and all those things. That's all basically happening because these individual atoms are trying to exchange their electrons so that they can fill their outer orbits. There's another thing you might have noticed is that they can share electrons. So they don't actually exchange them, but they get together and share. And you might have drawn a Lewis picture. You might have seen a carbon atom, right? Let me redraw that. You might have seen a carbon atom with these dots that show its electrons in its outer orbit. And you might have said, well, an oxygen atom could come along. Now, oxygen only has sort of 2 spots where it's not already full. And here's another oxygen atom over here with its electrons. And so what we say is they get together and they share these two, and they share these two. And this one over here shares these two and shares these two. So the end result is a carbon atom in the middle and an oxygen atom on either side. But the carbon is basically sharing, here's the carbon's electrons. Let me redraw that. Yeah, we draw the shared pairs the shared pairs as lines. I'm trying to erase that dot. There we go. So the shared pairs of electrons, which I drew in blue, okay, you can see that there's 2 for this one, and there's 2 on this one. So we draw them as lines like this. And then the oxygen atoms have the ones that sort of were left over that didn't get involved in this sharing at all. They were just sitting there. And then we have a name. We call this carbon dioxide, CO2. So now we have a molecule. And it's all because of all that chemistry of sharing electrons. Now, there are all kinds of combinations that you can make because you have so many atoms. You can combine them in so many ways. As long as it follows the simple rule of each one getting full, they can share in a billion different ways. So what this means is if you take a cup and you put a whole bunch of atoms in here, C's and N's and O's and H's and sulfurs, whatever. You put a whole bunch of atoms, you shake up the cup and you spill it out. What you will see is these little atoms will have gotten together in a whole bunch of different ways, making compounds that allow them to exist with full outer orbits. And there's so many molecules that you could make if you had, millions of each of the S's and O's and H's. In fact, for living things, carbon, hydrogen, oxygen, nitrogen are the most important atoms because 97 or 98% of every molecule that is inside of us is made only of these. Sometimes sulfur is thrown in there too because proteins have some sulfur in them. But even though there's, over 100 atoms, all you need are these to make living things, to make mostly living things anyway. Then there's smaller amounts of other atoms, like we have iron atoms inside of us, and magnesium atoms, and strontium atoms, and a whole bunch of others. But these are by far the most. So essentially, you put atoms in a cup, you shake them up, and they automatically make molecules because they want to fill their orbits. And we're used to some of these molecules, but you know what? There's bigger ones. There's molecules that are more complicated than just H2O or CO2 or even, barium nitrate. Let me give you an example. If you put carbon, hydrogen, and oxygen into a cup and shake it, then this can happen. This carbon would share with a carbon who shares with an oxygen, who shares with a carbon, sharing with this carbon, sharing with this carbon, who then shares with that carbon. It makes a loop. But it's not done. This carbon would share with another carbon. And this carbon would share with an H and an H and an oxygen who also shares with an H. And then we would have over here this carbon sharing with an oxygen, sharing with an H. And then another H sharing down here. And then an O, and then an H, and then an H. And you can see how complicated this gets. But it happens automatically. All you have to do is throw these 3 things into a cup. You throw carbon, oxygen, and hydrogen atoms into a cup and shake, and they'll make thousands of different molecules, but this is one they will make. It's one of the many possible ways that they could share. This molecule happens to be a molecule that's quite important to us. It's called glucose. It's a sugar. It's the sugar that's in our bloodstream, and it's the fuel source for our cells, right? Complicated. Now what we do, of course, is we like to make some shortcuts because these atoms get so complicated. We can't draw this every time. It's crazy. So here are some rules. You'll notice that there's a shape formed here, a hexagon shape, and it's formed mostly of carbon atoms. In fact, carbon is so popular that carbon is a shortcut. We make a shortcut when we draw carbon, and basically a corner means a carbon. So instead of drawing the hexagon like that, we take this hexagon with the C, the C, the C, the C, the C, the O, and the C, and we simply draw it like this, hexagon. We have to put the O in because it's not a carbon, but whenever you see a picture and you see a corner like this, or like this, or like this, or like this, or like this, those corners automatically mean carbon. So we don't have to write the C's anymore. Now we'll write some of the C's that aren't at a corner, but any C that's at a corner doesn't get written. Okay? That's one shortcut. The other shortcut is you'll notice that there's lots of H's attached to things. And H's are so common. And they attach, and in fact, when you put them in water, they attach and reattach and attach and reattach. So drawing all the H's is a pain too. So we don't draw them. We only draw sort of a line. And if you see a line, so a line that goes this way or this way or this way, if the end of the line like this has nothing on it, that means that there's an H on there. So now you'll notice we can redraw this shortcut. We can take our middle, which was the hexagon, like this. You have to put the O in because the O is not a C. So anything that's in the hexagon or anything that's on the picture that isn't a C for carbon, you can't leave out. So carbon's the only one that gets left out as a corner. Then what we can do is we can put little sort of ticks like this at each of the corners to show what else the carbons are holding on to besides holding on to themselves. And you'll notice, if we go back, there's something special. There's O and H's, and then there's H's. So let's put the O, and there's one here. Let's put those on first. Okay? Up at the top, we actually have a carbon atom that's not part of the circle. So he's up here, right? Now we could use our shortcut. We could just use a corner, right? So we'll make a line here, and then what we'll do is we'll put, let me just erase the one I drew there if I can, yep. And let me draw it a little bit differently. Okay, so we have a corner. Oh, I meant to draw it in blue. My mistake. Okay, blue. We have a corner, and then we could put a little cross up here. So see, in the middle of that cross, there's a corner. So actually, maybe red to show that it's a carbon corner was a better idea. Okay? So there's a carbon at every corner. All right. Now this carbon has an OH attached to it, so I'm going to write it here, an OH. And then over here there was an OH. Over here there was an OH. Over here there was an OH. And over here, I believe it was on this side. So you see what I've done? All those little circles. Oh, it was on top. Sorry, it was a different. This is called an isomer. So let me erase this here. Whoops. And let me put it where it belongs, on top. Because it could go on the top or the bottom, right? Depending. Okay, so here's our picture so far. So now we just imagine. I'm gonna show you where. See this spot, and this spot, and this spot. We imagine those, and even up here, this spot, and this spot. We imagine those are all H's, so we don't draw them in. So now we have a much nicer shortcut, right? So you can see these shapes form. Now you're going to learn all about this in grade 12, organic chemistry, but we need the shapes. We need to understand the basic idea so that we can figure out DNA. Okay, so that was glucose. Well, who cares about glucose? Let's talk about another molecule that can form if you shake carbon, oxygen, and hydrogen atoms in a cup. You can form a molecule called ribose. Ribose, which is the sugar. It's a kind of sugar we talked about in the last video. Here's what ribose looks like. It's carbon atoms holding hands in a pentagon shape. But at the very top of the pentagon is an oxygen atom. So each corner is a carbon. Okay? Then we have some arms here where more things attach. Now here's what attaches. I'm going to do some even more shortcuts. At the top here, there's a C with two H's on it, and an O and an H, all attached. So we shortcut it like that. That's a 2 on that H. There. All of that is attached. It's attached right to the C, right? And then on the bottom here is an H on the bottom of that line. Then we have, if I go around here, there's an OH attached to here, and there's an OH attached to here, and there's an OH attached to here. Okay. Now I also have a picture of this on the internet that I'm just going to, yeah. So here you can see it. It's written a little bit different than mine. Actually, if you look at this picture in the top right, in the black box there in the top right, you can see ribose. It's a 5-carbon sugar. Okay? So that's very similar to what we've drawn here. Now remember, there are hidden H's all around it, but that's the shortcut. So what we're going to do, even shorter, is we're going to just give this color blue, and we're going to say this is what we're going to draw for ribose. Okay? Now, what's interesting about this molecule is you could shuffle them around and get this, or you could shuffle all these atoms together, and instead of this OH forming here, you could get an H. So cross that out. If just an H holds on there, now you're missing that O, so it's called deoxyribose. Ribose with an oxygen not there. That's the only difference between ribose and deoxyribose. Okay? But we're going to draw them both as a pentagon shape. So when you see a pentagon shape, I want you to think of this ribose molecule or a deoxyribose molecule. They're both kind of the same. Okay. This is one of the parts that makes up DNA. Because, of course, DNA is deoxyribonucleic acid. So how do we get the nucleic acid part? Well, it turns out that if you take this ribose, well, I'm going to introduce you to another molecule, a molecule called a phosphate. A phosphate, you remember it as PO4 from grade 10, phosphate. And you might recall that PO4, and it has a -3 charge. Well, that's because phosphate is a phosphorus, and then what it does is it bonds with oxygens like this. So you see the 4 O's around it. And what happens is phosphate has H's that sort of bond on here like this to keep the oxygens balanced. But when you put it in the water, those H's can pop off. And when they pop off, you're losing 3 H's, and that gives you the -3 charge on a phosphate. So you got to remember that H's are kind of like the joining points of molecules. Anywhere you have a little H, it can easily pop off, and then you can join to something else, right? So we're going to draw phosphate in a different color. Let's use red. And we're going to draw it like a cross because that's kind of the shape it is, right? If you look at it, it's like a cross shape. So that's all. There's phosphate, cross shape. And remember that these H's can pop off and make it join to something else. And the same is true for this. These H's, wherever they are, they can pop off and let this join to something else. So H's are kind of like the joining spots. Okay, so there's 2 molecules, 2 shapes we have so far. Next, we have what are called nitrogenous bases. What are these? Well, they're complicated, and we're not going to learn to draw them all because I'm going to show you pictures instead. But we are going to learn their names. So let's use green for them. Their names are adenine, which we're going to use an A for. Then there's one called guanine. These were all named because of how they were discovered or found. And what's interesting is guanine was first found or discovered in bat droppings, which are called guano. So they called it guanine, bat poop molecule. Nice. Another one is cytosine. Notice how they all end in "ine"? There's lots of that in biology to help us remember all the families of molecules. Then there's one called thymine, which will be a T. And there's one called uracil. Now there are more. I've only drawn these 5. But these are the ones that are important for DNA and RNA. So there's more to this family of molecules. And you'll notice uracil doesn't have an ene on the end. So there's always exceptions. That's what makes organic chemistry so difficult. But here they are. Now, what do they look like? Well, rather than draw them, let's just have a quick look on the internet. So here is what guanine looks like. All right? You can see in the top right corner, it's got two sort of carbon rings. One has a 6-sided ring and one is a 5-sided ring, but it's also got ends. The reason these are called nitrogenous bases, right, is because they have nitrogen in them. So another word for these as a group are the nitrogenous, nitrogen-containing bases. So it's a complicated molecule, but it has little H's on it, which can pop off and join. So this can join to things. Here is what I think that was guanine. No, sorry, that was that guanine? Yeah, here's cytosine. Only one ring, lots of ends. You'll see that the ring has 2 lines on some sides. That means there's a double bond. Again, we're not worried about the chemistry. I just want you to see that these are actually things that are made of atoms. They're just clumps of atoms sharing. So when you draw, a big capital C with a circle on it, and you say, oh, that's cytosine, remind yourself that it's really just a bunch of atoms in a ring. Okay? Here's thymine. Again, nitrogen, a different shape to the ring, different things around the outside. Here's adenine, which looks a lot like cytosine. Actually, adenine looks a lot like guanine. Here's guanine. Here's adenine. Very similar. Very similar. They have 2 ring shapes, whereas cytosine and thymine are similar. See that? They have a 1 ring shape. And then there's this uracil, which is this one, which is also a 1 ring shape. So uracil and thymine are very, very similar. If you look at uracil, you can look at thymine, and all you'll notice different is if you look at the sort of at 10 o'clock on a clock, if you look at the carbon corner at 10 o'clock, there's nothing sticking out of it here on uracil, but for thymine, it's got a carbon and some H's sticking out of it. Remember, the end of that line is a carbon corner with H's sticking out of it. Actually, in fact, this one is just an H, I think. It's not a carbon at all. It's just an H. Actually, here's a better picture. There it is. So it is a C, sorry. It's a carbon with some H's. That's the only difference. So very similar. Thymine and uracil are very similar, which means they join in a very similar way. So that's the first thing I want to point out about these, is that thymine and uracil are almost identical. And so what happens with thymine and uracil is that they sort of occur. Oh, what have I done here? I may have lost my, I'm trying to go back to my original, here we go. Right, so I'm not sure, I gotta go here, there we go, sorry. So these two can be interchanged. So when you have RNA, then what we find is there's guanine in it, cytosine in it, there's adenine in it, and uracil in it. But when you have DNA, you have guanine in it, cytosine in it, adenine in it, but thymine instead, because these two are almost identical. So thymine is only found in DNA, uracil is only found in RNA, but they work exactly the same. So a U and a T are exactly the same thing, pretty much, and they do the same thing. So when you see a U or a T, it works the same. Okay, now, I want to point out something else that we'll come back to in a minute. Do you remember how some of them had sort of a hexagon and a pentagon sort of attached? They were 2 rings, and some were just really only 1 ring. Well, it turns out that, let me go back and show you here. Okay, so thymine and cytosine are 1 ring. Guanine and adenine are 2 rings. Okay, so remember that. So guanine and adenine are the 2 rings, and cytosine and thymine or uracil, right, those T and U are the same, are 1 ring. And it turns out that a 2-ringer can join lightly, very lightly, to a 1-ringer. That means that cytosine will bond, this 2-ringer or 1-ringer will bond to a 2-ringer. Now, it always bonds to the same one. It always bonds with guanine because of those different shapes. It won't work with adenine. However, the other 1-ringer, thymine or uracil, they will bond with adenine. So that means that a T always goes with an A. And this is important to remember, or a U. This could be a U as well. Because when we get into DNA and how it spells out our genes, this is critically important. Okay, so now, here's what we have. We want to build an RNA. So let's build RNA. How do you build it? Well, I'm going to draw this with pictures now instead of all the atoms. So we have a ribose right there. And then we're going to, actually, let me do it in colors. Let me erase that and do it in colors. Our ribose was the pentagon shape way up here, and we did that sort of in red. And then, oh, sorry, phosphate was red. So ribose is black, phosphate is red, and then green for the bases. Okay, for these nitrogenous bases. See, they have nitrogen in them. Oops, I just erased, sorry. Okay, an old man in technology. We don't get along very well. So let's take the black RNA, which is a 5-carbon pentagon shape. Then let's take our red phosphate. So what's going to happen is the red phosphate is going to join up with it, like this. One of those hydrogens can pop off, and this can join up with the ribose. And over here, hydrogens can pop off, and we can join up one of the nitrogenous bases. So let's say it's going to be adenine. So there it is. This makes something. Now what's cool is that this isn't it. This is just one tiny piece. But this chunk can be made, and of course you can make another chunk that has a cytosine on it, or you could make one that has a uracil, or you could make one that has a guanine attached to it. So the green parts can interchange. But the other cool thing is that this molecule can connect with another one. So right here, we can connect by popping off the H, and we can connect to another phosphate, which could connect to another 5-carbon ribose, which could then connect to another base. Maybe this time it's a different one. Maybe this time it's cytosine. And guess what? This can keep on going. This can then connect to a third one. And this third one, right, can connect like this, connecting to another ribose molecule. So all this connecting, right, all these complicated, and this one might have, say, now it could have another C, it could have another A, or it might have because it's ribose, it might have a U, a uracil. It can never have a T, and it can never have a thymine. And so basically, this process, ... can go on in both directions. So what you can make is a big, long chain, which we can draw simply as a line, right? So if you look at this picture, this part right here forms like a wiggly backbone. So maybe I should draw that line in blue. Let me get rid of that line. So it forms like this sort of wiggly line. So this line can now represent these shapes together, which represented all those atoms. And then what we have on the line are than the green bases that are attached. So in our case, we had an A, and then we had a C, and then we had a U. And this goes on forever. Maybe another A, and maybe a G, and then maybe another A. It could be anything. And of course, what you have now is a code, because the order of the C's, A's, U's, and G's is a code. You could have one RNA molecule that looks like this. And on it, it has A, C, A, G, U, U, G, A, C, G. But another RNA section or another molecule would have a different pattern. Maybe it's C, C, A, C, U, A, T. Oh, sorry, no T, my mistake. That's only in DNA. These are RNAs. So another U, maybe GG. And so you could build molecules, and just like we can build a word, so the word cat is an order of letters that makes us think of a cat, right? We've learned to call that cat. There's my cat. Well, these orders of letters, or orders of molecules or bases, can spell out what we call words, genetic words. So the order of the bases, the nitrogenous bases on the RNA molecule spell out coded words. They're encoded in the order of molecules on the base, or on the thing. Okay, so now DNA. Well, DNA is really almost the same thing. The only difference is it has a deoxyribose, right? So it has the deoxyribose molecules. Remember, down here there would just be no oxygen in that spot. So I'm not gonna draw that every time, but so here's DNA connected to a phosphate. It looks exactly the same. I didn't draw that very well, right? And then over here, nitrogenous base, say an A. And then down below, another one. There's our phosphate attached to another deoxyribose, which is attached to another base, say a C, which is then attached to, and on it goes, another, that C didn't come out very well. There's a C, cytosine, adenine. Okay, let's keep going. One more, just to get the idea. Now, here's the cool thing. DNA is more complicated. Let's put a T in there because it has thymine. Now, I don't know if I'm going to be able to draw this, because what happens with DNA, well, you know what I'm going to do? I'm going to draw that as a simpler version. Remember how we said we just draw this backbone as a blue line? Okay, so let's draw that blue line. Oh, there it is. And let's put the A, C, and the T on there. Adenine, cytosine, and thymine. Remember how I said that the adenine could form a weak bond with thymine, and cytosine can form a weak bond with guanine. And this thymine then would also form a weak bond with another adenine. And what if the three adenine, thymine, guanine over here were also attached to another ribose and another phosphate, which I can't fit in my picture? It would almost be like a whole upside down copy of the other side. And so that's exactly what happens. In DNA, the molecule is more complicated because two of them stick together. On this side, we'd have a thymine and a guanine and an adenine. And they stick lightly together because these things join. So DNA is actually a double strand, whereas RNA is a single strand. And the double strand is held together by the way that those, remember the two ringers and the one ringer, the way that they like to bond. So DNA is actually, it's almost like two RNAs stuck together. And then it also has the deoxyribose, and it has thymines instead of uracil. So see how complicated this molecule is. If we were to draw this using all the atoms and go back and draw everything the way we drew these, like this, even with the shortcuts, it would be a very complicated thing indeed. Let me show you pictures of some this already done. Here's RNA. So here's a picture of RNA on the right-hand side. You can see the phosphate cross in that sort of cyan-colored circle. You can see the ribose, which is kind of grayish, the pentagon shape. And instead of letters, they've put the actual molecular shapes in there for us, the C, the G, the A, and the U. That's ribose. Notice if you look at the ribose, on the bottom right, at about 5 o'clock, it's got an O and an H. Now, DNA, which I have somewhere, here it is. Actually, I think I made it. Here's DNA. On the left-hand side, you can see the phosphate, and in sort of with the yellow, then the orange pentagon, which is the deoxyribose. Notice at 5 o'clock on that pentagon, it doesn't have the OH anymore. And then you can see the adenine. But then you'll see there are little dotted lines coming out of the adenine on the right-hand side, which are joining to the thymine, which is, and everything is the same. The thymine is joined to another pentagon shape and another phosphate. It's all the same thing upside down. So this DNA molecule forms like, almost looks like a ladder, doesn't it? It forms like a ladder. And the rungs of the ladder are the inside, which are the colored parts. Those are the nitrogenous bases that are sort of attaching to each other. And remember, adenine always attaches across from thymine, and guanine is always found across from cytosine. So there it is, the DNA molecule. And because of this unique structure, this molecule has amazing properties. This molecule can reproduce itself exactly, so it can rewrite its own coded information. And it can use that information to instruct a cell on how to build one of the most important biological molecules, which is called a protein, which does 1,000 different things in biology. Proteins, they do everything. If you can make the right proteins, you can control how a cell will behave. So that's what DNA does. So now that we have a basic idea of what DNA looks like, DNA contains genetic messages, or words, whatever you want to call it, encoded in the order of bases. So let's draw a picture. So here's the DNA ladder. A, C, T, G, C. And across from it, we would have to have a T, an A, because remember, they have to join, A's and T's go together, and C's and G's, cytosines and guanines. And they would stick together like this to make the rungs of the ladder. And it turns out that because the ladder, because of the way the electrons are, this is complicated, it's got to do with electrons pushing and pulling on each other, the ladder gets twisted. So instead of drawing it flat like this, you'll often see the DNA molecule drawn like this, sort of with a twist. It's like somebody took the ladder at both ends and gave it a huge twist. And then you'll see these crosses drawn in here like this. Well, these crosses are the A and the T and the C and the G and the C and the G and the G and the C and so on, all the way down the molecule. The molecule is so big that you can have thousands of bases. Thousands of bases. Okay? Now, one more thing before we're done. The order of bases, let's say we have an order. Let's say we have on the side of our DNA molecule, we have A, G, C, T, A, T, G, C. Let's say there. And of course, this would be 10,000 long. You can imagine how much information you could store. We've discovered that each 3-letter combination of DNA is like a word. Each three bases forms a word. A genetic word. Now the word can mean many different things. The word can mean turn on, turn off, stop, go. Each of these words has a different meaning. But one of the main things is that each word, okay, the words are codes for things like on, off, stop, go, but also each code word is linked to an amino acid. An amino acid. So there are more molecules called amino acids. And some of you might know that amino acids are what makes proteins. If you're a bodybuilder, you might take supplements of amino acids to help build protein muscles in your body. Well, this word AAG might stand for a particular kind of amino acid, whereas this one might be a different amino acid, and this one a different. So what happens is that what the instructions of DNA really are is it's a way to put amino acids together in a certain order. And then when you put amino acids together, amino acid with another amino acid, which is different, and another amino acid, which is different, these amino acids make long, windy chains that can twist all around like this and join together and make a molecule called a protein. So essentially, DNA is instructions for making protein. That's what it does. And protein is a molecule that can regulate or control cell behavior in yet another more complicated interaction, which you'll learn a little bit about in grade 12 biology. Proteins regulate or control cell behavior. So proteins can tell cells to excrete things. They can tell cells to absorb things. They can tell cells whether or not those cells belong in your body or not. They can initiate immune responses. They can cause cells to become leaky and let things in and out. They can cause cells to seal up and not let things in and out. They can basically change the way cells work. And if you change the way cells work, you can control how they grow, what they grow into, and all of life. So proteins are the regulators of life. Proteins are made by combining amino acids, and the special order of each protein is different. A protein could have thousands of amino acids, hundreds or thousands even. Most of them are, I would say, are in the hundreds. But you can have big ones that have, thousands of amino acid combinations. And each different combination has a different shape that does a different job, and there's an unlimited number of possible proteins you could make. But the instructions for how to make them are encoded in the order of these base molecules that lie on the DNA molecule right here. So the order, that's what we mean by genetic information. And all of that is really just a bunch of atoms, all clumping together in a certain way because they share their outer orbits. And they fill their outer orbits. So can you imagine that the simple cell behavior, or the simple atom behavior of filling your outer orbits, leads to all kinds of combinations. One of the combinations happens to be a DNA molecule, and that combination has a code that can then instruct or store the information on how to build a protein, which is a molecule that can control cells. All right? So what we have there, just to sum up then, is a long chain of DNA, and it has words spelled as sequences of A's, G's, C's, and T's. Well, if you take several of these words, these three-letter words, and you put them together, they make amino acids, which can then be put into a protein. All right? So if a protein might be 200 amino acids long. So if you take a section of DNA, a whole section that has 200 of these three-letter words, all coding for an amino acid, then that particular protein chunk, that particular DNA chunk or section would make one protein molecule with 100 amino acids in it. What do we call this? This thing, this long piece of DNA that has all of the instructions required to make one protein? We call it a gene. That's what a gene is. A gene is a sequence of nitrogenous bases on a DNA molecule that spell out the instructions to make one particular protein. And of course, genes on a DNA molecule, because the DNA molecule is so long, you could have hundreds of genes all along here, hundreds and hundreds of genes on a molecule. Now, just to remind you, these DNA molecules basically all wiggle up sort of together, and they form chromosomes. So when we draw these weird things, chromosomes, this is a copied chromosome, right? It's basically this DNA stuff all wound up. And so inside a chromosome is DNA. On the DNA molecule, a certain little piece of it, maybe right here, that's a gene. And so on the chromosome, there's little spots all along that have genes, and that's why we draw these little lines. And we can actually, the DNA molecule is very difficult to see under a microscope, but we can see the actual chromosomes when the cells are dividing and the chromosomes have gotten thicker and fatter. So that's what it looks like. So you'll see pictures of chromosomes and you'll see a little shady spot like this, and they'll say, oh, that's a gene. It's called a gene. It's called a loci, or some people have said loci. I'm not sure. The problem with Latin is no one speaks it anymore, so no one's really sure how to pronounce it. But a loci is a location, a gene location. There's a spot. And what that means is if you were to dig down there on the magic school bus and get in close, you'd see a DNA molecule wound all up there, and there'd be a little section of it that would look something like this, a few hundred long, and that would code for the information to build this gene. To build this protein, right? The gene is the information that builds the protein, and the protein is all the sequences of amino acids that you would put together. Now, how do you get the amino acids together? That's for grade 12. That's the complicated biochemistry. But for now, we can understand the process. So that's what a gene is. And this took us, many decades after Mendel to figure out. Mendel knew